So if there is any issue in my presentation or voice, then please let me know. So coming to our first question. Okay, let's read it first. This autolysis during baker yeast production can be caused due to. So they are asking that when you produce this baker yeast, okay, yeah. So during the production, the autolysis means the breakdown of this product start occurring, and that is due to. So you have to give the reason why the autolysis of this yeast is occurring. So let's read the options. First one is absence of inositol. Second is absence of amylase enzymes. Third option is absence of pantothenic acid. And the fourth option is absence of biotin. So what happened in this? This pantothenic acid, due to the absence of this, like if this is not present, then autolysis of this baker yeast start occurring. So that's why for this question answer is C. Okay. Like you have to read, like first you are studying about the baker yeast production. So what you need, like what are the precursors, then what are the conditions required, like what is the pH, temperature and other requirement, like what are the other minerals or materials required. Suppose it require what is the specific enzyme that is required, why this pantothenic acid is required, or if any other component that is specific to every fermentation. So you must know all those things. Today's session mainly based on this type of questions only, because you have studied the fermentation of 3 4 products, so all the questions related to that only. Like this is the second question. Which of the following compound is are to be added with cane molasses during baker yeast fermentation process? So they are asking that if you are using the cane molasses, means another precursor is also used for this baker yeast fermentation. So they are specifically asking for cane molasses. So during baker yeast fermentation process, so what are the other compounds they added? So they have given the options like P2O5, MgO, magnesium oxide, calcium oxide, and none of the above. So you have used both this P2O5 and MgO, both are used in this cane molasses process. The percentage is fixed, like how much amount is used, that is mainly in between. Like for P2O5 it is 0.6 to 0.8% as compared to this molasses weight, like how much you are using that. So in proportion to that only this P2O5 and MgO are used, even though other component that are used in this process, that are also like their percent vary from this using the molasses weight. So both of the compound P2O5 and magnesium oxide are used in this different ratio. Okay, so for this question answer is both A and B. Now coming to the third question. This is like among the following of beet molasses and cane molasses, which ratio is more suitable for baker yeast fermentation? That previously we are studying about the cane molasses, what are the compounds required. So similarly beet molasses is also used for the fermentation. So they are used in a particular ratio, and this ratio is really important to make it more suitable, because this application of this baker yeast in different industry vary using this. Yeah. Okay, so you must know the ratio of both these type of molasses that are used. So for the baker yeast production, this is used in 60 to 40 ratio, like 60% of this and 40% of cane molasses is used. So for this question, answer is C. Okay, answer is visible. Okay, let's. This question is fermenter used for baker yeast production is constructed using. They have given that you must know that mainly this fermenter is produced of stainless steel. The only difference is amount of chromium that is present in different. So how much amount of chromium is used? Although answer is visible, so you must know that 12% of chromium is used. But any one of you tell me why this chromium is used in this fermenter? Like why we make the mixture of the stainless steel with chromium? Okay, any one of you? Okay, no one knows or no one want to answer. Okay, this chromium is mainly used to prevent this baker yeast from acid, so that it won't degrade by the acid or the pH. So that's why this chromium is used. Okay, so coming to the next question. Apart from yeast, which other microorganism is used during baker's yeast production? So you know that for bakery yeast production you use yeast, but what are the other microorganisms can possibly be used that we don't use normally in the industrial level, but that can be used? So what are the options? First one is acetic acid bacteria, and the second option is lactic acid bacteria. Then either both or none of the above. So what happened during this yeast production? This acetic acid bacteria, this is the major contamination. This is one of the major contamination that occur in the baker yeast. So this can't be used. This is the contamination. So then you have lactic acid bacteria. Yeah, they are possibly used in this baker yeast production. But preferably you use the yeast. Okay. So for this question answer is B. So this is the next question. Among the following, which can be added to prevent foaming during baker yeast production? So, like prevention of foaming is really important so that the amount of this baker yeast is maintained, and it also sometimes cause the contamination in that fermenter process. So we usually use the anti-foaming agents in the fermentation process. So for baker yeast production, what are the examples of that anti-foaming agents that we mainly used? So options given are: first one is tetradecanol, second option is tridecanol, third option is octadecanol, and D is none of the above. So for this baker yeast production, we use this tetradecanol and octadecanol as a foaming agent, anti-foaming agent, sorry. So they are added to prevent foaming. So for this question answer is both A and C. Now coming to the next question. Yeah, food yeast or nutritional yeast. So from the name itself, they are showing that this is a very nutritious, means when something that is used as a nutritional value. So what it contain? It mainly contain the proper amount of proteins, vitamins, other minerals also. So they are asking food yeast or nutritional yeast should have which of the following features. So they have given first option is high protein content, in which 40 to 60% weight by weight dry yeast. Okay, what does this mean? And second option they have given that vitamins of the B complex. That is also true. It means both the high protein content and the high this vitamin B complex is present in the nutritional yeast. So for this question answer is C. But what does this mean, 40 to 60% weight by weight dry yeast? So anyone want to answer this? Like what does this mean that 40 to 60% weight by weight dry yeast? Okay, you must know about this percentage weight by weight. Although you have studied few different like percentage weight by volume, percentage volume by volume. So all these are used for the representation of the amount. So in this means that weight by weight means if 100 gram dry yeast is there, then in that 100 gram 40 to 60 gram is protein. Okay, so this mainly representing the amount, that is the amount of protein present in the food yeast or the dry yeast. So that's why we are using here it in percentage of weight by weight, because they are telling both the things in the weight measurement. So for this question answer is C. Now coming to our next question. This is like the following microorganism does not create any problem during yeast fermentation. Okay, this is also a very simple question. What are the options given? That acetic acid bacteria. Okay, then second is mold, third is micrococcus, and D option is flavobacterium. So we recently discussed that this acetic acid bacteria is mainly used as a major, not used, it is a major contamination of the fermentation process. Especially yeast fermentation, this is a major contamination region. Then micrococcus is also similarly flavobacterium, it is also a bacteria that like occur as a contamination. So for this yeast fermentation, molds are the ones that does not create any problem in this yeast fermentation. So for this question answer is B, that molds are the ones that does not create any issue in the yeast fermentation. Okay, now this is your next question. They're asking that torula yeast is the microorganism used to produce fodder yeast. It is also known as. So they are asking the scientific name of this torula yeast. This torula yeast, and first one you must know that it is used in a fodder yeast, because that is also different. Some Fodder yeast means that are mainly used for the animal feed. Okay, so that is also require high protein content, high vitamins and the other components for their healthy diet. So this torula yeast is mainly used for the production of this. So what is the scientific name of this torula yeast? So options given are Candida utilis, Candida tropicalis, or both A and B, and the D option is Saccharomyces cerevisiae. So the torula yeast is also known as this Candida utilis. So for this question answer is A. That Candida utilis is used for the production of fodder yeast. Okay. So only for like all the questions you have seen are asking either about the microorganism, either about the media, either about the any other specific mineral or component that is required for its production, or what are the specific percentage of the precursors that are used in fermentation process. So all those things are required to maintain or to like when you are fermenting a product. So it should be cost effective, high yield, easy, or shelf life should be high. So we always try to make a product that has these properties and much more than that. So you always want to simplify the fermentation process. So for simplification you want to add different components, and also try to use the microorganism which will give the high yield. And also when you do any fermentation process you always try that the shelf life of the product should be high, so that it won't degrade or contamination won't occur. So this all those things, like to form these type of product you need to maintain the fermentation process using the different component, media, microorganism and other conditions. So you need to stabilize those conditions so that you will able to make a more product, because in this case you are studying all the production method at an industrial level. So when you make anything at an industrial level, then it should have all these qualities, because then only the industries will able to make that. Otherwise if it is not cost effective, suppose if any microorganism is making a yield of suppose yeast itself, but in a very high condition, very high amount, but that is not cost effective, so industries will not prefer that method. So you have to take care of all the parameters, and after seeing that then only you can try to optimize the process. So in the whole course you are studying the optimized process of the fermentation for making the different products that have different applications. Okay, for human consumption, animal consumption, for different industry, pharmaceuticals, cosmetics and other industries. So you must note this parameter properly. Now coming to the next question. Okay. During the fodder yeast fermentation, what is or are the byproducts? So they are simply asking what are the byproducts that are formed during this fodder yeast fermentation. So options are 5 days inosine monophosphate, second is guanosine monophosphate, third option is phosphate, and D is inosine monophosphate. So this 5 inosine monophosphate and guanosine monophosphate, both these are the byproduct of this fodder yeast fermentation. So for this question answer is A and B both. Okay, now coming to the next question. Which among the following is a source of nitrogen in the fodder yeast fermentation? Okay, they are asking which is the nitrogen source that you use. So first one is ammonium biphosphate, ammonium sulfate, or both A and B, or D, none of the above. So this ammonium sulfate is mainly used as a nitrogen source. Okay, in the fodder yeast. But can anyone tell me like why this is used, ammonium biphosphate? Or does this we use in the fodder yeast fermentation or not? Like what is the use of this? Anyone please answer this? Or either you can reply in message, like you can write it why this is used. Okay, now okay, now coming back to the question. This ammonium sulfate we used as a nitrogen source. Then this first one, this ammonium biphosphate, for which, why it is used? Okay, no one want to answer. Okay, this is mainly used for the potassium source. Sorry. Okay. Sorry, this is a phosphorus source. This is ammonium biphosphate. Okay, so for this question, answer is B. This ammonium bisulfate is used as a nitrogen source. Okay, coming to the next question, that which mode of cultivation is preferred for industrial production for spirulina species? First one is autotrophic, second is mixotropic, and C is heterotrophic, or all of the above. So this spirulina is also used as a feed product. So this is mainly produced as a auxotrophic cultivation. You have seen the pictures in your slides or in the lectures when you have seen that it is mainly a glass type of fermenters mainly are used so that light can come, and it's mainly a green color, means a chlorophyll is also present there, and the from sun it will take the energy, or from the light source you can say. So if you have like different shapes and sizes, these products, these fermenters are formed, but you have seen one thing, that they are made up of glass, means they are able to pass the light, so that light, they can take the light from the environment. Okay, So this spirular production occur in a auxotrophic cultivation. Coming to this next question, this self-shading is detrimental for spirulina cultivation. Which of the following can be a reason for self-shading during outdoor cultivation? So this is spirulina cultivation they are asking. So for this spirulina cultivation, this is mainly different shapes and sizes of the fermenters are used, and even both inside and outdoor cultivation is occurring. So you must know like what are the reasons so that this is self shading can occur, and self shading is not like good for its production because it degrades its so let's read the options. What are the options given? First one is depth of the tank increase to 12 to 15 centimeter. Okay. Second is the concentration or density of the cell increases. Third is when the solar lux increases beyond 30 to 40 k lux. And the D option they have given all of the above. So what happened when this spirulina cultivation is occurring, and when you are doing this in a like outdoor in the presence of light, so you try to make different shapes. Either it can be formed in tubes also, or you can form like connecting type of tubes. This well type of formation is also there for this fermentation. So what happened, this when you increase the depth of this by beyond 12 to 15 centimeter, like if the depth is like 15 centimeter or more than that, or even this much depth, like more than 10 to 15 centimeter, this is detrimental. Why? Because the spirulina present in that fermentation, it won't be able to get the light directly because other spirulina are present over the surface, so the sunlight is not able to penetrate till depth. So the spirulina present at the bottom surface, or like after 5 to 6 centimeter, the amount of light start getting reduced, so they are not able to get the light and they are not able to grow properly. So in the absence of light they won't be able to survive for a long time. So that's why the very depth, like tanks of very much depth is not preferred. So that's why if you increase the depth of the tank up to 12 to 15 centimeter, they will start self shading. Okay. The second option they have given that concentration or density of the cell increases. This also causes the same problem. Like suppose you have this only 5 centimeter depth of your fermenter, but the concentration of the spirulina is very high. Suppose this is showing the concentration. So it, if this is concentration is very high, then at the bottom, first thing is that again the sunlight problem, that if the concentration is very high, then it might not be able to penetrate, then it is also not able to provide proper oxygen and proper environment for their survival. So if the density or the concentration of these spirulina increases, then self shading also start occurring. And if the depth is start increasing, like or when you increase the depth more than 10 centimeter, then it will also degrade. Third option they have given that when the solar lux increases beyond 30 to 40 k lux. Okay, this is the like representing the solar light, that or the you can say energy in the form of solar light. So when this is like the optimum solar lux that is mainly required for this is 40 to 60 k lux. So this is the optimum required. So beyond this is beyond this, so this is not correct. If they have given that beyond 60k lakhs, then possibly this will occur. Okay. So for this question, answer is A and B both. If the depth is very much high or either the concentration is very high, in both the cases this spirulina cultivation is not occurring properly and self shading occur, means it will start degrading. So for this question, answer is both A and B. Now coming to the next question, which of the following method is are adopted for drying spirulina? Okay, so what are the options given? First one is freeze drying, second is spray drying, third is sun drying, and fourth one is cold drying. So for this spirulina drying, we used either sun drying, that you must know, and second one is a spray drying is also used. But in the freeze drying, what happened? You don't need to put it at a very low temperature. So that's why you don't prefer freeze or cold drying. Okay. For this spirulina, you usually prefer sun or spray drying. Like at a normal temperature, it will dry. So for this question, answer is B and C both. Okay, this is the next question. So now we are talking about the this high fructose corn syrup. In the last week, you have studied the fermentation of this also, high fructose corn syrup. So they are asking, this HFCS comprises of the following. What it will contain? Fructose, sucrose, glucose, and all of the above. So this high fructose corn syrup contain both fructose and glucose. Both these sugars are present in this. So for this question, answer is both A and C. But according to its need, like if HFC is used in a different industry, in a food industry, in a some other industry, the ratio of this fructose and glucose changes. Like sometime it can be used as is to 60 is to 40 ratio, sometime it is also used as 40 to 60 ratio, either 90 to 10 ratio. So this ratio will differ on the basis of application. But in HFCS, both of these are present. So answer is both A and C. This is important question and you must know the difference between this. Like what they are asking, match the amylases mentioned in column A with their cleaving site in column B. So what they are given, alpha amylase, beta amylase and gamma amylase. And what are the options given? Let's read. This alpha 1,6 glycosidic linkage, in addition to cleaving the last alpha 1,4 glycosidic linkage at the non-reducing end of amylose and amylopectin. Okay. Second option is alpha 1,4 glycosidic bond, but only on the non-reducing end of the polysaccharide chain. And the third option they have given is alpha 1,4 glycosidic bonds present in the inner part of the amylase or amylopectin. So that's why this is also sometimes used called as a endomylase. So first thing you must know that whether it is breaking the alpha 1,4 glycosidic bond or alpha 1,6 also. Like so what happened, this alpha amylase mainly break this alpha 1,4 glycosidic bond but at the endo part, means suppose this is the chain present. I'm making it very rough diagram, so don't go in figure of that. So this alpha amylase usually break like this alpha 1,4 glycosidic bond in the present in the inner part, not like at the corner one. This won't break this bond. Okay. Suppose this type of structure is there, so when you start numbering it 1 2 3 4 5, and this 1 6 1 carbon is present there in the form of CH2OH, so this is 6th. Okay, similarly in the second component or second monosaccharide, that 1 2 3 4 5 and the 1 carbon is present there, that is numbered as 6. So when it break this bond is called as alpha 1 4. This alpha and beta represent the orientation. So in this, alpha bonds are present. So if it break this bond alpha 1 4, this is alpha 1 4 bond, and if the bond form here with the sixth carbon, then the breaking of this bond is called as alpha 1 6. Okay, so you must know the difference between alpha 1 4 and alpha 1 6. This depend on the position of the carbon. Okay, so if this alpha amylase is mainly used for the, and it is also called as endomylase, means it is breaking the chain but present in the inner part, not the corner monosaccharide, it won't break. Then beta amylase similarly work, it also break alpha 1 4 glycosidic bond, but only on the non-reducing end of polysaccharide chain, means the carbon that the bond that it will break, it is the non-reducing side. So what happen, and this beta amylase, what it will do, suppose this is the chain, okay, it usually try to break the bond like after like two monosaccharides, so it usually forms the combination of, or it usually form the disaccharides after its cleavage, this beta amylase. Okay, it also break the alpha 1 4 glycosidic bond but at the non-reducing end, and usually it form the disaccharides. You must remember this. Then we are talking about the gamma amylase. What this gamma amylase do, it break both alpha 1 4 and alpha 1 6 linkages and at the non-reducing end of amylase. So what happen when it will break the alpha 1 6 glycosidic bond, suppose, just a minute, suppose this is the chain that is present. Usually the another chain that is like, suppose two chain are mixed, so they usually present at this linkage usually occur at the alpha 1 6 position. So it would also do this, that it will break, suppose this type of chains are present, this line form I am making, this is a chain of this amylase, so it also break this alpha 1 6 bond. Okay, so that's why gamma amylase is used for the breaking of both alpha 1 6 and alpha 1 4 glycosidic bond at the non-reducing end. Similarly, this alpha amylase is used for the endo, like, or the inner part, not at the corner one. And this beta amylase usually form the disaccharides. So you must know the difference of all these three, and the structure of all the three enzymes are present on your slide, that is very clearly representing there. So you must go through that. So for this question, answer is, let's see, first one is C. So option A is correct. For second is B correct. And C, gamma is used for both type of bonds. That is also correct. You can also study a little bit of about these, like what is the difference and what are the similarities in that. So like if we talk about the naming of these three, alpha, beta and gamma, if you go about the EC, that is EC is used for this enzyme, naming of the enzyme commission. So they usually name all the enzymes of the same family, like suppose this is 3.2.1.1. This type of naming is present for this alpha amylase. I'm not sure this is exactly, but this kind of representation is there. So they represent the family, then the subfamily, then which enzyme and which type. This one is representing here is alpha. Okay, similarly if you go for beta, that is 2, then 3. So you must know like what are the major difference and what are the similarities in the enzymes. And if there is any major enzyme that is used in various applications, then you must know it. Now coming to the next question. Okay, this is industrial amylases are produced from, that we are discussing from the very first day, that usually what is happening in the fermentation process, this industrial amylase is produced from this either bacterial or fungal source, or you can call it a microorganism, but as they have mentioned the bacteria and fungal, so for this answer is C. Why we don't use this plant and animal sources? There are two major things why we don't prefer this plant and animal sources, is that either this process of for using from plant and animal sources expensive, like it need more processing, and the second one is that it won't form in that much amount that we require, like we can't grow from animal source, you can't grow that large number of animals there. So when you feed the animal, it will cost much high as compared to the microorganism, and the amount that is produced is also low as compared to these bacteria and fungal sources. So mostly the industrial level fermentation process prefer the bacterial and fungal sources. Okay, so for this question answer is C. Now coming to the next and last question for this assignment. This is, which of the following statement is are true for fungal amylase? Okay, means we are forming amylase using the fungus source. So what are the options? First one is production is carried out by solid state fermentation. Second is the organism of choice is Aspergillus oryza. Third is wheat bran is used as a substrate. So for this fungal amylase, all the three statements are correct, means this wheat bran is used as a substrate, that is the initially this wheat bran is used. Okay, then we use the microorganism as this Aspergillus oryza. Then this fermentation type is solid state fermentation. For other for usually bacterial, we prefer different type of production that submerged, but for this solid state fermentation is preferred. Okay, so for this question answer is D, like all of the above. Okay, if anyone has any query, please let me know.
Ma'am, exam format, how it is going to be?
Sorry?
All of them are objective type or how many questions are going to be there?
In exam?
Yes.
Okay. Actually, I don't have any much idea about the exam because for this exam preparation there is another TA who look after all those things.
So, do we have any chance to interact with other TA?
Okay, for the exam.
Yeah,
Uh, okay, I'll try to inform them and ask about if they are able to join in this class for a little bit of time so that they
yeah.
will inform you.
So that will be helpful to us to prepare for the exam.
Yeah, sure. Okay, I'll try to talk to them.
Yeah, thank you.
Ma'am, I have a doubt.
Yeah,
Um, we are using Aspergillus oryzae for the production of amylase, na, ma'am.
yeah.
So like Aspergillus niger is also used, ma'am. Like Aspergillus niger.
For this amylase production,
Yes, ma'am. Is Aspergillus niger is also used for the production of amylase?
for amylase Aspergillus niger. Like I'm not sure whether Aspergillus niger is also preferred for this. Either I will check and let you know this Aspergillus niger because yeah they are from same family but because the enzyme production is different, so I'm not sure that niger species is also used for this amylase or not.
Okay, ma'am. Thank you.
I will
And
check
one
and
more
let you
doubt
know.
is yeah, sure. Yeah. And in this spirulina cultivation, na, ma'am, you're telling about that autotrophic condition,
Yeah,
right? Okay, so there are many ways to for the production of spirulina, ma'am. Like one of the method is open raceway ponds, right?
yeah, right.
Okay, so during like in open raceway ponds, this spirulina will be cultivated under autotrophic condition only, na, ma'am.
Yeah, that is also autotrophic condition.
Okay, and some other methods is also there, na, ma'am. Apart from autotrophic, many methods are there for production, na, ma'am, or only autotrophic condition. I mean, autotrophic methods only they will do.
Actually, this for spirulina because of the chlorophyll and the photosynthesis process, mainly we prefer this autotropic method for its production because it
Okay,
uses
ma'am,
the
because we have
photosynthesis process
heterotrophic and
mixed.
uh, yeah, mixed also we have. So
Yeah,
only by using autotrophic condition only we'll be
able to.
cultivating this
Yeah,
spirulina, ma'am, in open raceway ponds.
yeah, correct.
Yeah. Okay. Thank you.
Okay, okay. Any other doubt?
Ma'am, this is for assignment 10 question actually. Assignment 10, 10th question. Something like it's not very clear the answer is. Can you look into it and we'll discuss next week?
Okay, assignment 10 question.
Assignment 10 and 10th question.
Okay, I'll check it.
Yeah, please.
Okay.